What are just kind of like things that people are studying right now that you're very excited about?
There was a lot of excitement, and my group is very involved in this epigenetics. So we were talking about this strand of DNA being properly packaged.
Co-director of the Rochester Aging Research Center, she has published over 100 publications about longevity, sirtuins, and aging.
Vera Gorbunova, who's the endowed professor of biology and medicine at the University of Rochester in New York.
Her research is focused on understanding the mechanisms of longevity and genome stability, and on the studies of exceptionally long-lived mammals.
What negatively affects epigenome structures? Is it like diet, poor sleep? What?
DNA replication. Every time you replicate DNA, you have to open it up and put it back together. Every time a gene is activated to make a protein, it needs to be also opened up.
What does gene therapy look like? And then also, what are the risks that you mentioned?
Well, gene therapy, it's like you would take like a nanoparticle that would have a gene, Sirtuin 6 gene in it, and then it goes into your body and it starts making Sirtuin 6.
Do you think humans will ever figure out how to live forever?
Forever is a very heavy word. I don't think we are there yet or will be in any foreseeable future. But I think what we can do is to make people live longer and healthier, which is also extremely important. Who wants to live longer and be frail and decrepit? So the primary goal is to reduce sickness, to make people live healthier for as long as possible.
I see. So, I've been hearing a lot about the longevity space. You see a lot of the gurus kind of shine in the forefront now, and it's becoming a very big topic. What do you think the study of longevity will look like in, let's say, the next decade?
Well, that's a good question. Things are changing very rapidly. And right now we have some treatments that we can extend lifespan of various model laboratory organisms. And the next step will be taking these treatments and selecting those that are safe and effective in humans, and then starting trials in humans. So that's what I think the landscape where we are going, shifting now to taking some of the discoveries to practice. And of course, there is a lot more in terms of research that needs to be done. We have some ways of extending lifespan, but there is a lot that we don't yet understand. So there is a lot going on in understanding really the drivers of aging process and how we can target them.
Mm-hmm. Yeah, that makes a lot of sense. There's still a lot of questions to be answered. So what are some of the main ones that scientists are still trying to figure out? They're scratching their head, but if we really figure it out, it's going to make a huge impact in longevity research.
Well, so to put it in simple terms, because this is a very serious scientific question, right? Because there are many things we don't understand yet. But let's say many things go wrong as we age. We all know it. People can age very differently. Somebody has a heart problem, somebody has diabetes, somebody develops dementia. So we all age differently, and different system may cave in. But what's the underlying process? Because we all, even though we age very differently at different rates, but we kind of all end up in the same place. And at about the same time. Still it could be plus minus almost 40 years, right? If you think from people dying in their 60s to some people surviving to like hundreds. But still there are also similarities. So we want to find the underlying process that drives all of these pathologies as we get older. And we know some things. It's not like people have maybe multiple ideas about it, but which one is the most important? Is there one process that causes sort of kind of a central clock that ticks and then other things start to fall apart? And then if there is such one process, if we try to slow it down or even reverse it, will it reverse, other diseases of age? So that's really the biggest challenge. And scientists already discovered a number of things, like, for example, DNA in the cell. So our genetic information, it accumulates mutations as we get older. And mutations are bad. But then there is also additional layer here, because DNA, it's this very long linear molecule, like a thread, and it's very long in every cell. It's like, very, several feet of this DNA, and it needs to be packaged very neatly so that it's always accessible. And this packaging also gets messed up as we get older. And that by itself may also cause problems, because cell can no longer function properly if this, instruction manual is no longer as accessible as it should be. And so there are these various biological processes that are suspect right now, and we are trying to figure out, okay, which one, or a combination of some, which one should we target for the most clinical benefit.
Mm-hmm. I see. Yeah, that makes a lot of sense. There's a lot of different things that people are researching, such as, the clock, like you mentioned, such as, how people age and how different people age, and then also the DNA and how it can be structured properly. So out of all of those, are there areas where it's most studied, by many researchers because maybe they feel confident it's that one, or are researchers just kind of studying all of them, kind of relatively equally?
Yeah, well, people study all of them,
Mm-hmm.
and which is really nice. We need more people in this field. These are, very diverse questions. We know probably a bit more about, these are so-called nutrient sensing pathways.
Mm.
And these are the signaling pathways within the cell that respond to nutrients. So like there is food, you eat a hamburger, and then there are certain processes within the cells that start to respond to it. So it was discovered early on that altering these processes can slow down aging. And it was very well worked out using model organisms such as fruit fly or a worm, C. elegans. So you can perturb those nutrient sensing pathways, and then these organisms will live longer. So we kind of understand more about that. But when it's time to translate it into human health, there's still lots of questions, because that's where the idea of calorie restriction came from, which you probably heard about. That this is one intervention that extends lifespan in a variety of model organisms. In flies, in mice, you restrict their diet and they live longer, even significantly longer. But for humans, it's still an open question. Is it going to have the same effect? It was tried on monkeys, and monkeys live for a long time. I mean, not as long as humans, but still like 30 years. And the results were not clear-cut. So even in monkeys, the effect of calorie restriction, there were some benefits, but they were not as dramatic as in flies, for example, or mice. Because primates may be somewhat different. So even though we understand a lot how these pathways, we can tweak them and make flies live like 2 times longer, it's not entirely clear that we can do the same thing in a mammalian organism.
Is it safer, like when you're in the forefront of research, right? You kind of have choices of what to do. It's like, okay, do I eat less and be in a caloric deficit, or do I maintain my current calories, or do I eat more? And I know it's not clear, but would it be safer just looking at, the studies with animals? Is it safer to say, okay, well, maybe I'm just going to hedge and I'm going to say I'm going to be in a caloric deficit until we figure out a little bit more about, the nutrient sensing pathways?
Well, I think it's good not to overeat. At least with respect to humans, we know that often the problem is people just eat too much and not very healthy. So I would definitely encourage everyone to maintain healthy diet and not to overeat. And that is, I think we are all capable of doing it. Restricting the diet, especially dramatically restricting the way it was done for mice, like 30%, even 20%. It is very difficult for human to sustain it. And it could make people very unhappy if they not just don't overeat, like, okay, you feel you've had enough, so stop now, don't keep eating. But it's really like you have to go hungry, if it's 30% restriction, it's very severe. So I would say that realistically, it's not possible for an average person who doesn't really want to dedicate their entire life to this. So I don't even think it's a question, we can't do it. There were studies, people were very devoted to the idea of calorie restriction, and they would restrict themselves. But then when it was calculated, like what level people can do, if they have free access to refrigerator, people can do about 10%. I mean, more than that, it's like you have to be put in prison.
Right. I see. You have to be forced.
You have to be forced, right? So I don't think this is very practical way of doing it, and it would make people very unhappy. And at this point, we don't even know if it's necessary.
Right.
So I think sort of general recommendation would be, okay, just like go light, don't overstuff yourself. But I would not recommend people to, practice 30% calorie restriction. It's very difficult, and I think that would lead to a lot of frustration.
Yeah, I've been in the health and wellness space for quite a while now, 10 years. I love weightlifting, and that's one thing I noticed is if I restrict myself too much, I might do it for a period of time, but then eventually I would want to splurge, and and so then I start overeating. I have these cheat meals, and then it's just like this yo-yo effect. It's like I splurge, I overeat, and then I go back to it, and then I would want to splurge again. So I learned to just kind of have a optimal level of where I don't go too far in either spectrum.
Yes, and that may be, what works for you may be the best. There is also another concept of intermittent fasting, which is much easier for people because, as you just said, right, it's okay. Like you can skip a day even, but then if the next day you eat as much as you want, so that makes people healthy. I mean, not healthy, but at least happy and content with, their life. So that's an alternative strategy, and it was tried also on laboratory organisms, and it does bring benefit. There is life extension with intermittent fasting, sometimes just as good as with sustained calorie restriction. Whether the same concepts can be equally applied to people, we just don't really know. I don't think there were large enough trials because always when it comes to people, it's a question by how much you restrict and how strict you're going to be. Because if you tell, okay, people have to fast completely for a day, then it's just harder to recruit enough people to the study. So there were protocols, for example, people would eat just a salad for a day. And then the outcome of that study was that although there was some benefit, but it wasn't as strong as what's observed in mice that fasted completely for a day. But okay, that's no surprise. People were munching on salad. So we really don't know yet. But I would say perhaps if for someone who can tolerate intermittent fasting, that's a viable alternative.
Yeah, I've been doing intermittent fasting for over 5-6 years, and I found for myself it works really well. I know some people have tried it in my life and it doesn't really work for them. So I do agree with you. It's a matter of finding, what works. So as far as intermittent fasting, is there an optimal sort of hours of fasting at like 16:8, the traditional 16:8, or is there anything better out there?
Yeah, so that's also, another topic for discussion, because in the past, people thought that calorie is what's most important, and like, okay, you can choose whatever your target calorie number, and it doesn't matter when you eat. But recently we started learning that this is not true. It's very important when you eat. And there are all kinds of ideas, like some people would say, okay, I'm skipping breakfast, somebody else is skipping supper. And so still we don't really have a controlled study large enough to say what is best. But right now, again, based on mouse studies mostly, what can be said that it's important not to eat during the nighttime, or at least when it's dark, because our circadian rhythm is extremely important. And combining maybe very mild calorie restriction with also eating at the right time of day, which would be anywhere, during daytime, has much greater benefit than either one of these interventions taken alone. Whether to skip breakfast or supper, there is really no scientific evidence to say which is preferable. Some people were very quick to start skipping breakfast just because it's easier. People, also culturally, I think in the United States, people often don't eat breakfast. They just jump, with a cup of coffee. But then there is some epidemiological evidence that it's not that good, that actually you want to eat breakfast, but then don't eat heavy dinner. So, lots of ideas, but there is really not enough evidence for either one of them. So again, I would say whatever works for different people, to choose a regimen to just sort of minimize the time span when you eat. Don't eat too early in the morning, don't eat too late at night. So I think breakfast at a reasonable time is okay if it's, again, not at 6 a.m., but like as late as it fits your schedule, and then no very late dinners. Better to finish your meals while it's still light outside.
Yeah, and I do adhere to that. For me, I've tried a few things. I've tried 3 meals that, within the 8-hour window. I've tried 2 meals, and then I've tried 1 meal to the extreme. Actually, the 1 meal I did in like an hour, 2 hours, and that was too extreme for me. And so I found that my optimal level was within 8 hours, kind of like to your point, during waking hours. And it's easier for me to just skip breakfast and go for the lunch and the dinner. And so, that makes a lot of sense, that as long as you don't eat too early, too late. And is there any research? I've heard that if you eat too close to your bedtime, to give a few hours of fasting or not eating anything before bedtime to enhance sleep. Are you aware of any of studies?
Yeah, so there is, I think there is enough evidence to say that eating very late and close to your bedtime has lots of health problems with it. Even, besides longevity, people get like heartburn and reflux, and I think in terms of blood sugar control, it's not optimal to eat very late. So there is lots of reasons to not to eat late at night.
Right. So for you, are you doing intermittent fasting? What's kind of like your regimen?
Well, for some time I was, again, it just the way my schedule worked, I was not eating lunch because I would come to work and just work my day and then come home and eat dinner, and it would be kind of disruptive for my work schedule to have a big lunch in the middle of the day. And I pretty much maintained the same routine, that I eat breakfast and a very light snack in the middle of the day, and then I eat dinner. And I'm trying to eat breakfast not early and dinner not late.
Yeah, awesome. So besides, time restriction, are there any other things that are being studied for, working with the nutrient sensing pathways and altering it?
Well, I think these are currently two major, calorie restriction, intermittent fasting,
Mhm.
and then time restriction. So that's probably the most we learned. Of course, you can pharmacologically manipulate those pathways. And one intervention that was very consistently showing benefits in model organisms is rapamycin, which is inhibitor of mTOR signaling. And it does extend lifespan in mice and flies. In humans, there are clinical trials going on with different results. There was a trial looking at aging of immune system, and from a small trial, it seemed that it gave benefit. Their immune response was getting better, but then in a larger trial, it didn't replicate. So we still don't know. Rapamycin is not an entirely benign substance, and this is why it's not an over-the-counter thing. People do develop certain side effects from it. So again, it's not something that I think people should all jump on, start taking. People also try to come up with a regimen that is, the best, because rapamycin inhibits proliferation of cells. And if you're thinking about various processes in the body where you need cell proliferation, you don't just inhibit. So people come up with ideas, okay, like you take it maybe once a week, and then you let your body recover, and then you take it. So there are all sorts of protocols, but again, we don't have one evidence-based regimen that we know works very well, and people get all sorts of side effects like mouth sores and other things.
Mm-hmm. Yeah. So, I noticed that, we there's still so much that we don't know. What are kind of like the things preventing us from not knowing? Is it like there's not enough patients volunteering to be in these studies? Is it not enough funding? Is it just time? The more we study this over a long period of time, we're going to figure it out eventually. Like, what are some of the barriers, that are preventing us from reaching all these discoveries?
Well, it's a very difficult problem, first of all. I think that's what makes it not so fast.
Mm-hmm.
In biology, things often take time. It's an incremental process. Of course, we need more funds. We need more people in this field. And experiments are getting more and more expensive, because if you're thinking now about a long-term study with humans or with mice, it all costs money. So funding is important. Getting volunteers, yes, that's also difficult. And, looking for strategies or like treatments against aging, there are also sort of policy-making issues that slow it down, because if you are looking for a treatment for a disease and there is a very clear outcome, and you can show, okay, the drug improves outcome, and then FDA would recognize it, and insurance would start paying for this drug, so that incentivizes pharma industry. But with aging, it's more difficult, because if your outcome is what the person lived 2 years longer, or like some biomarkers didn't change as fast, it's not clear whether insurance would even agree to reimburse it if it's an expensive treatment.
Mm-hmm.
And that makes pharma less interested in pursuing it. So it's more, I think right now, it's still more within academic research, because here, okay, we can get taxpayers' money to develop this. And then if it really works, then the hope is, well, if then the government would say, okay, we want to reimburse people for taking it, because then everybody's healthier. At that point, perhaps pharma would also be interested, but right now, not yet.
Right. Yeah. There's no profit incentive
Yes.
yet. Yeah, it's funny how that works. So, you mentioned the length of a study. How? I mean, you, I think uh, just not being able to. I mean, you probably wouldn't be able to study someone for their whole life, right? Because it's just too long. What's kind of like the optimal amount of time for a study?
If you're talking about human study for
Yeah.
mice, well, it typically takes 3 years.
Hmm.
If you want to really see how they all age and die. For humans, yeah, that's a problem. We live very long. And it makes studies very difficult because it may be, well, lifetime of a researcher. But a big progress in this area was achieved when people developed biomarkers. Those biomarkers are not perfect yet, so it's a big effort to improve them and develop better ones. But at least we can somehow measure aging process and say, okay, these people, maybe the aging slowed temporarily when they were on the treatment. But still, that takes time. So I would say 1 year would be a minimum. Maybe 6 months. There are some studies with 6 months, but sometimes it's inconclusive. So I would say at least a year is needed.
Right, at the minimum.
Ideally longer, right? But
Yeah.
then get into these very long studies.
I see. So what are some of the current, I mean, you're in the forefront of the longevity research. What are just kind of like things that people are studying right now that you're very excited about? If there's some promising results and people are excited, like what's kind of making the buzz currently?
Well, there are all kinds of ideas. There was a lot of excitement, and my group is very involved in this, in epigenetics. So we were talking about this strand of DNA being properly packaged. And that seems to be maybe the underlying cause of aging, because if this code for life is now becomes inaccessible or messed up, so that may explain loss of functionality in tissues and multiple systems level. So there was this realization is kind of becoming more generally accepted that loss of epigenome structure and stability may be the underlying cause of aging. And then, okay, how do we address it? So one way of dealing with it was to apply what's called partial reprogramming. I don't know if you've heard this term. So basically, the idea is kind of old because evolution has used it. Like every time a new child is born, the child is born young, right? 0 age. The child is not born with the age of their parents. And why does it happen? Because you would think, well, you take a cell from mother and father and make a child, so why the child is young? Because they both had old cells. Even the eggs and sperm were around for a while before they conceived the child. And the secret is that there is some process of rejuvenation that is happening very early during development or during the formation of the egg. There is some kind of rejuvenation during this process that rewires everything. The DNA, everything is rewired, and now you start from new. So the idea was, how can we use this process and now apply it not to the eggs, but apply it to our somatic tissue, to an adult tissue? Can we kind of use the same strategy, whatever rejuvenates a newborn child or a fetus, and apply it to our somatic body? And the idea was that there are transcription factors, they are called pluripotency factors, that were discovered some years ago, that if you activate these transcription factors are activated in early development in the embryo. And if we take these factors and put them into like an old cell, you take a skin cell, you put these factors in it, you can make an embryonic cell. So it turns back the clock. But you don't want to turn it back too much, right? We want to be ourselves. We don't want to become some embryonic mass. and that was the biggest challenge, because if you drive these factors full force, it just makes your cell, back to the embryo, and we don't want that. So here is that would be reprogramming, but you can do it just a little bit partially, and this is partial reprogramming. And it had some success. So you can put these factors in like the whole mouse, turn them on, and some mice will die because they reprogram too much. They become kind of an embryonic body. The tissues don't work anymore. But some mice will survive, and those mice can actually be rejuvenated. So that was a breakthrough, because you can now actually take this evolutionary force of rejuvenation that we all use, but we give it to our next generation, not to ourselves, and then somehow apply it to somatic body. But it's very far from being practical, because, as I said, like many mice die. You have to give it just enough. But it's a concept. So at least, it's possible to turn back the clock. It's possible to rewire epigenome. So something that we are studying is how to do it very mildly and safely, and not necessarily by using these pluripotency factors, because they are too strong. They can turn you back into an embryo, and you don't want that, because really embryonic undifferentiated cell within a human body, it's cancer, right? You don't want to turn yourself into a tumor. You want to just rejuvenate very slightly. And so that's why we don't want to use pluripotency factors. We want to use some other factors, but that can sort of fix the DNA a little bit without turning us into an embryo or into a tumor, which is even worse. So one of such factors is called Sirtuin 6. It's a protein that deals with DNA packaging. So it can come back, package it, when it all unravels, it can come back and package it again. So we are studying this Sirtuin 6. We know that if you put an extra copy of it into mice, they live longer, and it's safe. They don't turn into tumors, nothing bad happens to them. So now we are looking at various ways, like gene therapies, how we can enhance it. Also pharmacological ways to upregulate, to turn it on a little bit more. And one treatment that we are very excited about, and which is very safe, so I have no problems recommending it to people. We found that seaweed has a molecule in it that can activate Sirtuin 6, which may have something to do with why people in Asia, or Japan, South Korea, people live longer. They eat a lot of seaweed. At least I could say, well, there is no harm in eating seaweed.
Right.
Everybody can eat as much seaweed as they want. And we are now trying to kind of understand how this molecule works. How can we make it work even stronger? And we also now looking to maybe do a more controlled clinical study with people. We would give them seaweed, see how their aging biomarkers change.
Wow. So what? So seaweed helps turn on Sirtuin 6.
Yes.
And that will help, with keeping epigenome structures and repackaging. So are there any other foods? I can hear some people saying, ooh, seaweed. Any other foods like seaweed that could help turn that on?
That particular molecule, it's called fucoidin, is mainly in seaweed, in brown seaweed. So it's not the one that is a sushi wrapper, but this, seaweed that people make soup with it or stew. Kelp. So kelp is a species of brown seaweed and has a lot of fucoidan. Now, there are other foods that are associated with longevity benefits, and not because of sirtuin 6 necessarily, but people studied the effects of blueberries, other wild berries. In general, they have molecules that seem to be beneficial, inducing various beneficial effects. So that's, I think, again, you can't go wrong with eating blueberry.
Right. So seaweed, is there something special about seaweed? Is because it's under the sea, or there's more good bacteria because it's under the sea? Like why seaweed in particular?
Well, we don't know why it makes this molecule that we see very strongly activate sirtuin 6, and sirtuin 6 we want to activate because it's linked to longevity. Now, why exactly is it doing it? We're trying to understand it more from perspective of biophysics and biochemistry, understand how that happens. So we don't have a full picture yet, but sort of why seaweed has such benefits, like we don't know. It may be just random. It happens.
Right. Is it something that could be made in a lab, like for drugs or like a supplement that people take?
It could be. It's easy and cheaper right now to just get it from the sea. But it could possibly be made in the lab.
Got it. Okay. So why, you mentioned epigenome structures. Why does, or what affects it? What negatively affects epigenome structures? Is it like diet, poor sleep? How can people prevent it?
Everything, life affects it because our cell goes through rounds of DNA replication. Every time you replicate DNA, you have to open it up and put it back together. Every time a gene is activated to make a protein, it needs to be also opened up. So there is a lot going on. So it's just the effect of life. Of course, various unhealthy things like radiation, different smoke, various DNA damaging agents are bad, definitely. But even if you do all the right things, there is still some damage that is happening just from within.
I see. So there's some damage that is just totally outside your control, right?
Right.
Okay.
It could also may have something to do even like with circadian cycles that we talked about, because there are some time of day when DNA gets repaired. So it may be when people don't get enough sleep, or they're disrupted, their circadian cycles at night are disrupted. That may also affect repair of DNA and epigenome. So these are all things are tied together.
So sleep is a big one, making sure that your circadian rhythm is on point. Are there any other really major big ones that people just really need to be aware of?
Well, no, I would say these are probably we have the most evidence, because we know that people that work at night shift, there is very clear health detriment. People develop more cancers, they age faster. So yeah, I'm trying to think what other sort of obvious things. But it's mostly lifestyle, even stress, right? So excessive stress, psychological stress is also linked to various epigenetic effects as well. But again, this is something we all know, right? You need to avoid stress as possible.
Yeah, stress is a big one. That's one thing I noticed is self-awareness is huge, for knowing that you're stressed. I feel like a lot of us kind of go through life just in a wheel that we're not even aware sometimes that we're stressed, that we're showing stress signs until our health is affected by the stress, then we're like, okay, well, I need to like chill out more. But yeah, I sometimes think about that. Like we go through life just unaware, and so just having that self-awareness when you're stressed, the signs that you show when you're stressed, and then just being aware about it allows you to actually do something about it afterwards.
Yes, that's true. Also, again, these are sort of more psychological things. Social connectedness is very important. Most centenarians or people who live past 100, they are very well rooted in their communities. They have good family structures. Oh yeah, that's very important.
Wow. Awesome. So besides consuming fucoidan, are there anything else that is looking pretty promising to maintain good epigenome structures?
Well, so there was some talk about vitamin B12, but that may be too early to say. I think, we need more evidence. I would say at this point, fucoidan is, I think, pretty much the only one intervention that would be directly targeting epigenetic structure. Of course, we are also looking at gene therapy approaches for SIRT6, which would be more powerful, but also there are more risks associated with gene therapy, but that's something we're working on. Also with fucoidan, what we found that not every isolate of fucoidan will activate SIRT6 the same. It may have to do with like different seaweeds. It's like, different grasses. They're not all the same. So there are different plant species. And we find that some of them activate, some of them don't. So that's something that we are testing in the lab, different batches of fucoidan. And we are trying to understand which specific structure is the best for activating SIRT6. But I would say, other than that, I don't think there is kind of a easily accessible way of doing it. Of course, there are people working on developing these pluripotency genes and maybe targeting them directly, but there is nothing yet that you can do yourself.
I see. That's interesting. There's different types of fucoidan in seaweed, so you want to make sure you're consuming the right one. Is there anything that you for sure, like, oh, it has to be organic, or it has to be,
we run biochemical assay in the lab where we have SIRT6 and we mix it with fucoidan and we see if it activates it. And we work with Do Not Age company, so we test their batches, and then they can say, oh, well, this is not just fucoidan, this is actually activator of sirtuin 6. But other than that, I don't know, because we really don't know at this point what particular species of algae. Sometimes even like they would tell you, okay, the species, but you don't know where they harvested them, in which ocean, and there may be also different conditions how these algae were growing that may affect. So the only way we know right now is just our biochemical tests that we do in the lab.
Got it. So you mentioned gene therapy. What does gene therapy look like? And then also, what are the risks that you mentioned?
Well, gene therapy, it's like you would take like a nanoparticle that would have a gene, sirtuin 6 gene in it, and then it goes into your body and it starts making sirtuin 6, which, would be very effective if everything works as expected. So right now we are testing it in mice, and we want to see that we'll get the same benefit, that it is safe. We expect it to be safe because we know that if you genetically engineer mice to have extra copy of SIRT6, they're actually very healthy. It doesn't create any problem, but gene therapy, it's still sort of developing technology. It's already approved for some life-threatening conditions, like when people are born, for example, missing some immune gene, and like they need to be, they're need to live in a bubble unless they get gene therapy to fix that gene. But it hasn't yet been approved for something like just augmenting your lifespan. But I think that may happen fairly quickly if we can show benefit.
I see. Okay. So I heard in some of your other interviews, you mentioned the importance of NAD and how sirtuins, NAD is needed for sirtuins. So can you talk a little bit about NAD and its link to sirtuins?
Well, so sirtuins use NAD as a cofactor. So they're enzymes, and for enzymes to work, they need another molecule. Like they would take a cofactor. They can take, for example, sirtuin 6 can add ribose, like a ring of sugar, to other proteins, and it needs to take this ribose from somewhere. So it uses NAD. It takes ribose from NAD and puts it on a target protein. So it needs NAD. Now the question is, by consuming NAD, are you going to activate your sirtuin 6? And we don't actually know that because if other sirtuins, for example, sirtuin 1 is very sensitive to NAD. Sirtuin 6 isn't particularly sensitive. Like it needs some NAD, and it's actually very good at binding to NAD and using it. And it doesn't really matter if you give a little bit more, a little bit less, it will still bind what it needs to. So that's why I don't think NAD supplementation alone is going to have significant difference in activating sirtuin 6. We really need to find specific activators for this protein.
I see. So it's kind of like it's a tool that sirtuins use, but if they don't have that, like if you increase that, it's not going to really help that much.
Yeah, that's right. It's not like, okay, if you don't have that, it's probably a problem.
Right.
But we always have some in our cells, and for sirtuin 6, that's usually enough. So it's not that we don't have enough sirtuin 6 activity because we need more NAD. Sirtuin 6 is good at using whatever is available. Whether NAD can benefit by activating other sirtuins, there is a lot of research in that area, and there is evidence, there was some good evidence obtained that it may help muscle. So for muscle aging, there seems to be benefit. For overall lifespan extension, not really. There are many studies that didn't see benefit for lifespan extension. So I think that is still the area where there is no consensus at this point.
So sirtuin 6, there's a number 6 on it. What are the other numbers and how many sirtuins are there?
Well, there are 7 in human. And they were just numbered in the order in which they were cloned. So the number doesn't have any particular meaning attached to it. Sirtuin 1 was the first one discovered and studied, so it was called 1. It doesn't mean it's the most important one. Based on the effects on aging phenotypes in mice, when you delete the gene, the most interesting aging phenotype has sirtuin 6, with sirtuin 6. With sirtuin 1, you can, depending also on the mouse strain, you can delete it and have absolutely no phenotype, or you could just have dead mice. So it's a little bit less connected to aging from that perspective. But with sirtuin 6, if you delete it, you get mice that age very fast. And their DNA is very disorganized. But if you give an extra copy, they live longer. And that's not really the case with any other sirtuins.
So it is the most important one out of all the
I would say for aging, yes. They're probably all important for some biological processes, but for aging, I would say sirtuin 6 is the most important.
Right. Have people tried cutting the other ones out and just having sirtuin 6 by itself?
No, I mean, you need all of them. I mean, they each do something in the cell, but with the others, it's not like you give extra and you live longer.
Right. Uh-huh. I see. Interesting. So out of everything that you do, I mean, what is your longevity protocol? Like tell me about your diet or your sleep, how many hours you get, do you exercise, tell me about all the things you're doing for yourself to increase your longevity.
Well, I'm trying to exercise. I don't exercise very vigorously, but I try to walk at least, a couple of miles every day. Often I take my dog for a walk in the morning and in the evening. So, to stay reasonably active, I like swimming. Again, we talked about diet. So yeah, not overeating. I love fruits and vegetables and seaweed whenever I can. Just trying not to eat too much and avoid, these heavy carbohydrates and like grain products. I mean, I eat a little bit of grain, but just I know that it doesn't make me feel good, so I'm trying to minimize it and eat more vegetables and fruits instead of grains. Sleep, that's a difficult one because I'm just I don't sleep very well. I'm trying to get enough sleep, but it's not always possible. But I think it's a good idea to at least have 7 to 8 hours.
Got it.
And I'm trying, also with the to only eat during daytime. I don't eat at night ever.
Got it. What's kind of like your cutoff time, like 5 p.m.?
No, that would be, of course, nice, but no, it's more, I would say probably 7 or 8.
Okay. Awesome. And when you exercise, how often are you exercising?
No, every day, but, moderately.
Moderately. Yeah. That's cool. And then as far as, foods, you mentioned fruit, vegetables, seaweed. A couple things about that. Are you eating like organic fruits and vegetables or?
Yes, I'm trying whenever possible. And this is, I think that is not just longevity, it's a general health recommendation because there are so many pesticides and herbicides that we don't want in our diet. So of course, organic vegetables is the best. And I completely avoid anything that has food colorings in it or various preservatives. Okay, sometimes, of course, if you are at a function and you just eat whatever is put in front of you, but whenever I'm in control of what I'm eating, I would not eat anything that has food color, preservatives and stuff like that.
Right. So earlier you mentioned berries. Are there any like specific types of fruits and vegetables you're really targeting, or is it kind of a wide variety?
Variety is good, but I like blueberries, raspberries, and blackberries, all those sort of more wild berries, because there is some thinking that all of these plants that are more on the wilder side, so they are not as susceptible to, for example, pests and insects, because they're producing various molecules to ward off those pests and diseases. And the same molecule are like spices for the same matter, like they have these molecules. And when humans consume it, they can induce mild hormetic effect on you. So we are very good at dosing ourselves, like with a little bit of spice and herb, because what gives herbs their aroma, these are again those molecules that are maybe evolved there to ward off insects and fungi diseases. So that, not sometimes it's difficult to exactly isolate, okay, what is the compound, although we have some, different polyphenol fractions and like whatever gives color to blue color plants. We know that those compounds have benefits, but also it's a combination, like everything together in the plant, in the fruit. Strawberries are also good if they are organic, because those that are not organic, there is too much pesticide in them. Another thing that people don't realize, but I think it's important to avoid the artificial sweeteners, because people would think, oh, I will take Diet Coke, and this way, I'm not consuming sugar. And this is a very bad idea, because artificial sweeteners are really bad.
Right. And then the seaweed, is there a particular brand that you're pretty confident about, or you just have done research in and you're like, I'll stick with this one?
Now we are testing for do not age companies. So we tested it in the lab. We gave it to mice. We know that our mice live longer if they eat it. I mean, this is not to say there are probably other fucoidants that are just as good, but you just never know, because we see about 30% of batches activate SIRT6 and 30% don't.
Wow. Okay. Wait,
I mean,
you said
70% don't.
70% don't, okay.
Yeah. So that's what I could recommend for fucoidant. But again, if you just get some seaweed and make soup with it, that's also good, and you can't go wrong.
Gotcha.
Maybe it won't activate your SIRT6 as much, but it's healthy.
All right. So just to wrap up here, what are, if you have to summarize, we talked about so many different topics here, but if you have to summarize the most high impact things that people can do for their longevity, what would you say in addition to eat seaweed?
Yeah, eat seaweed first. Second, healthy diet full of fruits and vegetables. Eating at the right time, moderate exercise, keeping good sleep routine, and taking care of your circadian cycle. Another thing that you can disrupt your cycles completely is when people, something wakes you up at night and you turn the lights on, and it doesn't take too much for your brain, like all the receptors in your eyes, to then think, oh, it's daytime, and it will change everything. So if you have to get up at night, you have to try to keep the lights very low.
Awesome. Thank you so much for being on, Dr. Gorbunova. This has been a real pleasure. And I'm sure a lot of people learn from you. How can people support you and the work that you do?
Well, I'm always open for suggestions, so people can write to me. You can Google me easily. My email is on the web, and I'm always open to different ideas, collaboration, support, whatever.
Awesome. Any particular link or place people can go to learn more about what you do that you want to promote?
Oh, well, there is a website for my laboratory, but if you just Google Vera Gorbunova aging, it will bring you there.
Awesome. Great. Thank you so much. Thank you, everybody, for listening, and I will see you in the next episode. Bye now.
Okay, thank you. My pleasure.